So thank you very much for having me here for this presentation. And today I would like to share with you our some new technologies we developed to really unlock the single cell resolution information from FFPE samples. And as this is a cancer center, I'm very sure that you know about the current bottlenecks and also the FFPE value of FFPE samples. But before I jump into all that, probably just a very quick overview of our company for those of you who don't really know about us so much. And Singleron was founded in 2018. And at that time, high throughput single cell sequencing was still relatively new technology and not very much mature. But my co-founders and we already saw the value of single cell sequencing not only in research but also in clinical translation, like to really decipher maybe very complex diseases like cancer, and also identify the more effective therapeutic strategies than what we have today. So as of now, our company has really been growing in the last 8 years, and thanks for all the support from our collaborators and also our clients, and of course the hard work of our teams. And currently we have clients in more than 30 countries, and we have more than 800 publications from our customers. And like in more than 1,000 institutes, we work together. Our products are used in more than 6,000 labs now. Because we think to enable users to use the high throughput single cell technology more effectively, it would be very important to have an end-to-end solution. Starting from dealing with different sample types, because especially in a clinical research and later on clinical translation setting, you deal with all kinds of different tissues, biopsy, and of course also blood samples. Sometimes if you do research, you also need to deal with for example, organoid or other sphenoids or whatever, like all those different sample types. So our workflow and also our product portfolio really starts from very beginning to get high quality single cell or single nuclei suspension from different sample types, so that those single cells or single nuclei can be further processed for high throughput sequencing. And once you have data, like the raw data, it's one thing, but another important step would be really to analyze those data and try to get the most insights out of this kind of high complexity and high dimension single cell data. So we also have a cloud-based data analysis platform, as well as a database to help people to really get insights, not only the raw data, but also insights from the data. So the principle of the technology, I have a chip here, but it's been troubling with me too much, so it's heavily scratched. If you like, you can still have a look. And it's a very simple principle. We have a microfluidics chip with about 250 up to 250,000 microwells. And then we use Poisson distribution. If we load the chip with, you can pass around. And then if you load it with a suspension of cells or nuclei, they will disperse based on Poisson distribution. So then in each well, you have either no cell or like one cell. That's how we separate them. And then we use our barcoding beads to barcode each cell in the well once the cells are lysed. And we capture, therefore, their genomic information, and then we do further processing to sequencing libraries, and we do and in the sequencing, like the RNA or other analytes we would like to sequence, they would have the unique cell barcode. That's how we could tell which information is from which which cell. So after that, we get like the information, high throughput single cell information from thousands of, tens of thousands, hundreds of thousands of cells, and each at like single cell level, and in each single cell we get the gene expression information or other information from thousands of the genes. And then with this background, I would also like to remind everyone here, but you are experts for cancer research and other research. I think you know this more than I do. The current bottleneck in cancer as well as other diseases. Like cancer is, if we live long enough, everyone, like a huge percentage of us would eventually probably develop some kind of cancer. And from all the cancer patients, actually, although we have target therapy, we have immune therapy, we also have cell therapy, and we have now also the very popular ADCs, and we also have now, I think, the this kind of nuclear therapy for cancer patients. All those new therapies, they are quite advanced. They really increased the survival of cancer patients in the last decades. But still, if you think about the overall cancer treatment of all those advanced therapy, each of them, including the immune therapy, the majority of the patients probably don't respond so well to the therapy. And also, even if they might have the initial response, the cancer almost always comes back. And how could we use, like better predict the therapeutic outcomes of those patients, and also develop maybe even more effective treatment modalities that we don't even know today. Some new technologies and also some new ways of measuring the tissue, the cancer tissues and cancer samples might be needed, so that if we understand the cancer and the tumors better, it would then, as scientists, it would enable us to really find the more targeted and more accurate ways to treat them. So that's where Singleron, as well as maybe a lot of researchers in this room, also believe the high throughput single cell sequencing information. If you could, cancers are always very heterogeneous. If you could really have their detailed information on genes, on epigenome, on gene expression, on proteins, and so on at single cell level, this would enable us to identify maybe the minor signals that could be very significant in the detection as well as treatment of cancer that are masked by if we do bulk analysis. So that's also what we keep working on, and developing new technologies and products that we believe would help people in this direction. And as cancer researchers and clinicians, experts, as you might know, FFPE samples or FFPE method has also been a very commonly used way in the clinical setting in cancer. So before I go to the next slide, I would like to ask very quickly, does anyone know how many FFPE samples we might have in the world currently, all over the world? Anyone would like to take a guess?
More than 2 billion.
Yeah. Another guess? And another question would be, when do you think the method was officially established in the clinic? FFPE method, to use FFPE to preserve tissue samples and use it for a clinical examination.
18th century?
Yeah, that's a little bit early, although I think at that time people already use some preservatives to preserve, for example, birds and animals. But anyway, here is an overview. I also just found those online. yesterday. I think it's quite interesting also to know that the method was officially established for a clinical setting about 130 years ago. And before that, of course, people already used formaldehyde to preserve maybe also some plants, animals, and so on. And it's estimated that there are more than 2 billion, I think, with the speed we are generating right now, like about 100 million per year. I think we would reach the 2 billion very quickly. And of course, if people now feel there are methods to help them to analyze also FFPE samples more effectively at molecular level, maybe the motivation to use FFPE, this old method, might even be higher. And but how about single cell analysis? We probably, I think you probably also know some of the bottlenecks and challenges the FFPE samples pose if you would like to do molecular analysis of them. Let me see whether it's the, yeah. Okay, so because you use formaldehyde to preserve them, this is a chemical reagent that could really also cause modification of the nucleic acids, especially if, like, later on this block of FFPE sample is just put in the lab somewhere, exposed to light and oxygen and all those environmental things, and also degradation in the process, and would also be a problem. Like, you don't really have intact DNA and RNA anymore. You only have the pieces, because in this process of treatment, processing, and later on the storage, the DNA and RNA would often get degraded. And yeah, so all those really contribute to the fact that if you want to analyze FFPE samples, first, the extraction of DNA and RNA is really not always so easy. You don't always get high quality DNA and RNA, and they might also have chemical modification. That's also why, like in NGS, there are a lot of algorithms to correct this kind of chemical modification, like false positives or negatives caused by those chemical modifications. And also, because of the degradation and chemical modification, if you would like to sequence them, you need to generate them into like sequencing libraries. Those biochemical reactions are not always so effective if you have to deal with like small pieces of the DNA or RNA of heavily degraded and modified pieces of them. So this problem we probably already know from even the bulk sequencing of FFPE samples. And for single cell, you really have to get information from tiny little one cell. That could be even more difficult if you are talking about getting information from like picograms of RNA or DNA from each cell. So there have been some methods trying to solve this problem to generate single cell RNA expression information. For example, this is from 10x Genomics, the Flex product, as you probably, some of you probably also know. And they use a hybridization-based method. So they design a panel about 20,000 different probes that can anneal to different genes inside a cell. And through the hybridization of the probes, at the end, they could do ligation if there is a target. And at the end, by sequencing those probes as proxy, you would be able to tell like which genes you have expressed in those cells. It's a nice method. I think it did circumvent the problems of the FFPE bottlenecks. But this is, in my opinion, just like a quantification analysis. It's a little bit like single cell qPCR, because you know like how good or how much the genes are expressed. But it's not really sequencing, because with sequencing, we would probably like to have new discovered discoveries, you would like to see, like, for example, for cancer, whether certain mutations are expressed there, or whether there are other genomic variants that are there. You would like to use the NGS method to really make new discoveries instead of to count how many copies the gene is expressed. So we think, so that's why we try to develop our own methods. And I told ChatGPT yesterday to our technical principles, and this is what the ChatGPT came up with. It's not 100 accurate, but I think it's quite good. So I'm using it with credit to ChatGPT. So what we do is you have a fragmented RNA in the FFPE cells or nuclei. They don't always have the poly A tail at the end of the 3' end. And also they are very difficult if we would like to use the typical oligo-DT priming method to generate cDNA. So what we do is we use random oligos to first. So first we fix the cells or nuclei. And if it's from FFPE, it's already fixed. So basically the cells or the nuclei, they are already permeabilized. You can already push the enzymes into those cells or nuclei. So what we can do is we do first nuclei extraction from the FFPE tissues, and then we incubate them with enzymes and primers. So those enzymes and primers can now get into the nuclei because of the fixation and also the permeabilization. And then we have this kind of in-cell RT reaction based on random priming strategy. So first, all those fragments with or without oligo-DT sites, they will be randomly primed and they will get the cDNA. But that's not all. We also, in the pod of single cells, we also have this poly A addition step, so that all the cDNA, after they were reverse transcribed to cDNA, they will bring this poly A tail. So this would enable them to be captured by our barcoding beads when we do the single cell library generation. So then after like this step, so nuclei extraction and also the reactions inside of the nuclei, we can just load those nuclei suspension as we do normal single nucleus sequencing to our chip, or if you have a machine, to the instrument, the cartridge on the instrument. And everything else is the same as our standard single nucleus library prep procedure. But because of the random priming, you get the mRNA sequence not only from the 3' for the quantification, you also get it to cover most of the gene body of each and every gene. And with that, you can detect expressed variants, potentially also fusions and isoforms. And if you are looking at maybe inherited cancers, you can also look at a new specific expression of SNPs. And additionally, we can also detect non-coding RNA, including microRNA from those cells. And any questions? Yeah, so this is basically, again, explanation of the procedure. Like you get the FFPE samples, we extract the nuclei. We can do it manually or with our tissue dissociation instrument. And after that, we have this RT reaction in the cells and RT and tail addition. And after that, we just do everything based on the standard single nucleus protocol. And yeah, it can be done manually in this chip on our instrument. And the list is growing, and this is what our R&D has been already tested successfully. And the tissue types from, like, we did most of the mouse tissue types in our development process. And we are also gaining more experience with different human sample types, mostly also cancer types. So also some examples of how the data would look like, because of course you would like to have also good data quality from FFPE samples so that you can make the right studies and right conclusions from the sequencing results. And as you can see here, those are different mouse tissues we have tested, and with relatively average sequencing depth of between 20,000 reads per cell to up to 40,000 reads per cell, which is standard range. Depending on the tissue type you test, we could get between, I think the lowest was 1,627 genes per cell to up to 3,387 genes per cell. So it's actually quite good quality and very probably even a little bit better than the oligo-DT priming, because here, based on the random primer, we do capture more parts of the mRNA as well as those non-coding genes. And if you happen to have a not so good FFPE samples, this is like one example where the DV200, meaning like the pieces larger than 200 BP, is only 17.3%. So it would be almost excluded from FFPE normal NGS reaction based on the quality. And here we could detect with the random priming and tagging method, we could detect more than 3,000 gene per cell. So yeah, even if you probably have very old FFPE samples that are not looking so great, it's still worth giving it a try. And we could also have relatively good coverage from the 5' to 3' of the gene body, meaning that if you have, for example, KRAS mutation on the 5', the chance of detecting it is also high. Yeah, and also the non-coding RNA, because non-coding RNA's importance has been revealed more and more for different diseases. And as you can see here, this is a mouse bladder cancer sample we used. We can detect different types of non-coding RNA. And we grouped them based on the species of non-coding RNA. And of course, if you have certain non-coding RNA in mind, you would like to look, you can also easily look at its expression in different cell types. And we can even, here is again, if you look at specific non-coding RNAs, you really can see they are expressed quite differently in those different cell types, and you can really do more in-depth studies. And we could also, this is quite interesting actually, we ignored mRNA totally. We just used the differential expression levels of different non-coding RNAs to cluster our cells. And we could see that we could do the cell type clustering quite well if we only looked at the non-coding RNA. And then we also tried to do annotation, and we could also annotate the cell types quite accurately based on the non-coding RNA expression levels, meaning that in all those different cell types, the non-coding RNA, they are really also expressed with a certain pattern. And it's really worth now with the availability of the single cell analysis, single cell resolution, it's really worth to take a deeper look at those non-coding RNAs at single cell level, which was not so always easy previously. And we also analyzed, for example, some very similar cell types, like T cells and NK cells in the microenvironment, to make sure that even if they are like expressed at small percentage in the whole cell composition, and they express a lot of similar genes, we could accurately separate them from each other by looking at the gene expression levels. And the reproducibility, here we have 4 different mouse lung samples, and we ran 4 plicate, and we saw quite similar also the gene expression patterns and clustering. And and also, of course, we would like to also use the real-life FFPE samples that have been processed and also stored there. And so here we took 4 mouse bladder cancer FFPE samples. They have been stored for more than 20 months. And as you can see here, so first with all 4 samples, we got good results with relatively, let me see, where is the sequencing depth, relatively low sequencing depth from 30 to 90 G. We could get more than 1,000 genes per cell, and up to more than 2,000 genes per cell, because there the sequencing depth from this one sample is higher. And so this sample, although the DV200 is also lower than 50%, but still we could detect more than 2,000 genes. And if you, so those 4 samples, they are from 2 groups, 2 are control mouse tissues and mouse cancers, and 2 are treated with a certain drug. And what we could see here is, like first, you get quite similar pattern from those 2 controls versus those 2 treated samples. And also we see consistent results of, for example, increase of fibroblasts as well as mononuclear phagocytes upon treatment, and also decrease of the cancer cells upon treatment in the mouse from those old FFPE samples. And we could also look at the difference of like non-coding RNA expression patterns upon treatment in macrophage, in T cells. Of course, you can also look at other cell types and other non-coding RNA if you are interested. And for isoform detection, because we have the almost full-length information from the mRNA, we could also, by looking at like the reads of different junctions, we could also get the information or deduce the information, like which kind of isoforms for those genes we might have in which cell types. And we also got some real clinical samples, not only mouse samples. And here is a human bladder cancer, 3 FFPE samples. And we extracted nuclei, we did library prep and sequencing. As you can see here, it's also relatively moderate sequencing depth of 14 to 30,000 reads per cell. And we could get for all those samples more than 1,000 genes. And if we have like 30K reads per cell, we have more than 2,000 genes detected. And again, despite the lower than 50% DV200 scores. And also the, sorry, if you go back 1 slide. So you can also see clear separation of different cell types that can be annotated accurately. And if we go to the next slide, that's like the QC results we got from a real life customer with their human liver FFPE samples. It's even better than what we generated ourselves in the lab. And as you can see here, the mean reads per cell is 27 to 45,000 reads per cell. And we could get especially this one sample is really good. We got almost 3,000 genes with 27,000 reads per cell with a very low saturation of 12%, meaning that if you sequence further, you can also get much more information. Okay, so Just in conclusion, we think we have a quite good FFPE single cell sequencing solution that enables you to detect not only the gene expression levels of different mRNAs at single cell level from FFPE samples, but you can get also the real sequence information. Additionally, you can also get the non-coding RNA information from the same cells, as well. In addition to the mRNA information, you can also, with this kind of almost complete gene body coverage, you can also deduce the isoform information, fusion information, and so on. And we do think this could be a very useful tool to help cancer researchers to really disclose all the secrets that have been previously locked in, like the treasure of the FFPE archives. And we would also like to take some challenge. As you see, like so far, I think most of the FFPE samples that we have been testing, they are a couple years old. If you have really like very old samples that but important samples that you think would worth a try, we would like to give it a try. I was even thinking maybe we can find in some very old archive, some famous person's sample, FFPE sample from 100 years ago. Maybe we can even give that a try. And and then let people know what exactly this one famous person had. Maybe he or she didn't even had the cancer as people at that time thought. But anyway, so there might be some interesting projects I think we can do together. And I would also like to go through very quickly our spatial solutions, because for cancer, I think it's also very important sometimes to not only know which cells you have in the FFPE samples, but also the relative locations of those different cell types. And we are introducing a service of spatial transcriptomics, and later on also spatial multi-omics. It's based on the DebitSeq technology from Rong Fan, our co-founder from Yale University. And what we do is, again, also like you have a slice of the tissue. And with this microfluidics chip, you can really pump into the cells on this slice the barcodes, as well as the enzymes to process them. So instead of using chip or droplet to somehow, like using mostly, I think people use chip to with specific like oligos and specific locations to have the location barcodes. Here we really use this fluidics methods to barcode each cell on the slice. And with that, we do have the flexibility that first, this is also like sequencing-based technology. And we use both random primer and RT to reverse transcribe RNA. So just like our FFPE single cell sequencing product, we generate the real sequence from the full RNA. So you get the full length RNA information. You also get the non-coding RNA information because of the random priming. It's species agnostic. So if you have some experimental animal for cancer model that's non-human, that's non-mouse, you can use it without any problems. We don't rely on any pre-designed probes. So this would be like a service only, while for now, the FFPE product would be like both a kit and service. And we also have some experimental data. For example, the median gene per spot, we could detect this also in this case for mouse brain more than 3,000, because we really have the free floating primers in the cells, so the RT is more efficient. And here is also the annotation of the mouse brain, and you can also detect different non-coding RNA in the mouse brain from the same slice. And yeah, and here is intestine, where you can also have nice clustering of different cell types based on the transcriptome data. So if you are interested, you can feel free to ask my colleagues to get more information. And thank you very much for your attention. And yeah.